site is the exit site. Right? So it's about to exit, so exit site. So I would say RNA processing would work as well here. You could actually argue.
We're cheating whoever put that.
Yeah. Give yourself 1 billion points.
So I really got first place. I'm still in first place. Boom.
Each amino acid would take 3 codon, 1 codon, 3 nucleotides.
What? Oh my god, I went from first to sixth. What a downfall. I realized after the fact because we've been talking.
So the promoter region is where the RNA polymerase is binding. Transcription factors are really the factors that recruit the RNA polymerase and allow it to bind, kind of like the activators in a sense.
I love it.
So post-translational is basically saying after we make the amino acid sequence, after we make the polypeptides. Remember, polypeptides are formed. Some are formed on attached ribosomes. Attached ribosomes, the polypeptide goes into the wealthy yard, where modifications occur. Travels to the Golgi apparatus, more modifications occur. Kind of go way back to the beginning of science. With that, so random reason. You got us. Okay. So those modifications that we talked about could be like the adding of nitrate, adding of lipids, right? Those are the modifications. Maybe we cut some of the amino acids out. So they're all post-translational. The adding of the amino acids is actually translation. And by I.
Yes! Boom! Something I'm coming for you. Who is first place? Oscar. Snagit. So much.
That's me.
APE. A site's where the new one comes in. P site is where the existing transfer RNA with the growing chain exists. E site is the exit site. APE, yep. Okay. What was the last?
We just really digested down briefly before that. We did a project. Already skimmed through it.
Did the menu project.
Yeah.
I never talked about this. I'm not going.
It's that. I
It's not really a big thing of but it's kind of interesting. Here's what's known as nucleotide wobble. So as long as the first nucleotides match up, that third nucleotide in the codon sometimes doesn't necessarily register, and it codes for a different amino acid. It's known as wobble. I left it there because it's a funny term, and whatever.
Jesus, it's so alive.
Touche.
Yes, first place.
First place. In first place.
Yes.
Especially when it's Texas the song.
Texas sauce.
The song that goes with Texas. Or
You memorize that.
sugar, phosphate group, and nitrogenous base. Right, the amino acid's not part of it. So the exon are the coding sections of the RNA transcript. They're kept. And it's mRNA. Remember, the messenger RNA is what's actually being coded. The anticodons are found on tRNA. They ensure the proper transfer of the amino acid. The codons are on mRNA. Don't make that mistake. SNRPs just mean RNA. SNRPs, SNRNPs are just RNA. Single nucleotide ribonucleic proteins. It's on the. I'll look it up. It's on the PowerPoint. Small nuclear ribonucleic proteins. So you don't really need to know SNRPs, but just know, like we talked about spliceable why it was special because this protein component, there's also an RNA component, and the RNA component has enzymatic qualities. That's what it's referring to. This is the hardest question.
You're like,
Don't make the easy mistake. How many people make? Oh, 4 people made the silly mistake. DNA polymerase makes DNA, not RNA. The taxa box is unique to eukaryotes. Prokaryotes don't have
I did. I was 7th place and snatched your spot.
We didn't really go into eukaryotic regulation, but we're going to highlight that the taxa box is a eukaryotic thing, not a prokaryotic thing.
Oh my god, I snatched your spot.
Prokaryotes do have promoter regions. I just don't necessarily have the taxa box. Just make sure you're okay with that. So prokaryotes do have promoter regions where RNA polymerase binds. They just don't necessarily have the taxa box. Where does translation occur?
Oh!
Who put the? I do want some of these. I feel like. But we're learning, right? When we make mistakes, we learn.
Especially when you explain.
Good. Did I need to explain to us, or we're okay? Transcription occurs in the nucleus. Translation occurs in the cytoplasm because the mRNA leaves the nucleus. Docks with the ribosome.
It's unibrow.
universal genetic code, right? So universal. Formation tester bonds. So what process showed? It was transcription that was being shown, right? Because transcription is a process of changing the DNA message into RNA. That was transcription. No esterification. Good. And translation is the process of doing the actual assembly of the polypeptide, which happens after that. Spicy splicing? No. All right, so although RNA processing technically could work for this, the answer is not RNA processing. All right, so all this is talking about is the RNA splicing process, and it's just showing how we're cutting out different regions. So we're showing alternative splices. That's all it means by alternative splices. So nothing really too different, but alternative splices. Was that the last one? Oh, no. Figure it out. So, a couple things. You need a codon chart to do this. You will have a codon chart. So memorize codons amino acids. Secondly, I initially read it and I thought, oh, the answer must be easy. It must be the first one because the start sequences is always AUG. Yeah. But however, that's not a codon sequence. It is the what? It's the tRNA sequence. So to do this question right, you would really have to code back to a codon. So AUG is the start codon. So therefore, it knocks that one out. And then we need the codon chart. We really need a codon chart to do this.
I memorized AUU, but like,
yeah, honestly, on my test, I won't try to trick you like that, and they won't do that in the exam either. They're not going to try to trick you by giving you a TRD. Well, I've never seen it before. I don't think they would do that. But they would always give you a codon chart if you have to do something like that. And you'll have a codon chart tomorrow. RNA is a nucleic acid with needed protein. That's simply not correct. All the other statements are correct. I did mention this briefly. So it is just the enzyme that forms or that joins the amino acids that transfer RNA. So it's the amino acyl tRNA synthesis. So which one requires energy? All the above. Now, if you got rid of the parentheses, ATP, you would knock translation out of it. Translation uses GTP, but just kind of grouping them together. I never really mentioned their names. No, you don't need to memorize their names either. So Beetle and Tatum. Michelle Beetle and Channing Tatum.
I know, I thought that was like a joke.
So universality is just kind of the term you would use because it's a universal genetic code. Is not a function of the 5' cap. So that's the biggest thing. You know, it definitely, we talked about the main part is the degradation, but it also acts as like kind of the lead to lead the nucleus. It also helps to identify the front of your RNA molecule to bind to the ribosome instead of the back end. So all those are really kind of functions of it. Again, the main function is to prevent degradation. But however, that 5' cap does not code for anything because it's not AUG. It always starts with AUG. So it does not code for any part of the protein. We're done. Who's first place?
First place.
Oscar?
Someone who made first place.
Give it lots of sad faces. Or happy faces, either way. Angry.
So excited.
They're so that first place.
2.7.
Wah, wah, wah.
That's our anniversary. My Italian or